As Dean of the faculty, it is my great pleasure to thank you for your belief in the importance of research. Whether you've supported the faculty's research fund or a specific research project that's meaningful to you, every donation makes a tangible difference to improve the health and well-being of our families and friends and the broader society in which we live. Thank you.
My name's Dr. Grace Lidgewood. I'm an Early Career Research Fellow here in the Department of Anatomy and Physiology at the University of Melbourne. Alzheimer's disease is a progressive neurological disorder that causes death and dysfunction of the cells of the brain called neurons. And what this ends up causing is a decline in cognitive function in the patient, so their ability to socialise, to function independently. It's the most common form of dementia, and what we know is that with our ageing population, by the year 2060, the number of patients with Alzheimer's is projected to double. I'm a retinal biologist, so I'm really interested in the cells of the retina, which is the light-sensing tissue at the back of the eye. What we know from clinical studies is that the cells of the retina become affected in Alzheimer's disease. And this is perhaps not surprising because the eye is a physical extension of the brain, so it's part of the nervous system. One of these nasty pathological hallmarks of the disease is a protein known as beta amyloid. And what this does is this becomes really sticky and forms a plaque on the outside of neurons, and it causes the neurons to basically die. And I'm interested in looking at the retina as a surrogate model for Alzheimer's disease. I work with a particular type of cell called a pluripotent stem cell. We can make retinal cells in a dish, and essentially what this does is it creates a patient's very own tissue in a dish. So we can start to look at some of those features of Alzheimer's disease in the retina in real time. In the last couple of years, we've had some really exciting breakthroughs in our project. What we've been able to show is that our retinal cell models mimic a lot of the pathological processes that happen in Alzheimer's disease. Our cell model was able to make beta amyloid, which is the sticky, yucky protein in the disease that causes the neurons to die. And we saw this in much higher numbers in the Alzheimer's lines compared to the controls. To our knowledge, this is one of the first studies to show this effect in a retinal cell model in the lab. The support of donors is absolutely critical to these kinds of projects, these blue sky, really discovery-based projects. On a personal level, support of donors has been transformational to my ability to be a scientist. And I just want to say thank you so much because it's honestly been the best thing that's ever happened for my research career.
I'm Richard Tottill. I'm a research scientist, an academic, and I lead a research group in the Department of Clinical Pathology and University of Melbourne Centre for Cancer Research, and our group's called Rare Disease Oncogenomics. Fundamentally, all cancers are driven by acquisition of mutations in cells. Something that we've been doing is looking at what we call genomic pathology, really using genomics to enhance our understanding for diagnosis and also treatment of disease. And what we understand now is that cancer is not just one disease. We've got many different diseases based on where they come from in the body, but even those diseases can be split into multiple disease types. And so understanding this allows us to diagnose the cancers better and to apply different treatments in a more precise way. An area that really excites me right now is this area of liquid biopsy. What this entails is really looking at DNA that's being released from tumour cells in a patient into the peripheral blood. This allows a really minimally invasive method to sample the tumour and really alleviates the need for a tissue biopsy. This can be done serially over time, and then we can monitor the patient's genome or the tumour genome, looking for potential resistant mechanisms, also just a recurrence of disease over time, allowing a more early intervention for treatment of that patient's cancer. Philanthropic donations are very important for our work. Often these projects can't be funded through established agencies. We're trying new methodologies, new techniques. Perhaps we're starting research in a completely new area that may involve a rare disease, for instance. It's critical to have that early investment in the research that will then make these projects viable, giving us preliminary data to make us competitive for further grant funding. I really, really appreciate all the commitment and the donations that have been given to our research.
I'm Carlos Opaso, a Chilean translational neuroscientist. I have been working in schizophrenia since 2015. We have been trying to tackle the mechanisms of schizophrenia. We found that there are some protein problems in schizophrenia. And the next question was, what could be explaining this? In the case of schizophrenia, we found that iron is accumulated in a particular part of the brain, this is the frontal cortex, where all our main functions are located. Usually at the age of 14, 15, people start feeling that they don't belong, and they feel alone, and they get problems to adapt, which impacts the studies, in the school, in the university. But then they have other type of symptoms that they are called negative symptoms, where they withdraw basically from society. And also later on they have problems related to cognition. So it's a complex disease, and therefore the individual, I believe, that feel very lonely and isolated. So hopefully we can help them in some way. I'm not saying that we have the cure or the treatment now, but we are trying to take care of these biochemical signs that we have found in the brain of these individuals that may help. There are some pharmacological treatments that they are not 100% effective. For the cognitive and the negative symptoms, there is nothing available. And we believe that it's a possibility to target this excess of iron and treat some of the symptoms. Science is expensive business, but the benefits are clear. The donor has transformed our possibilities to continue with our research in difficult times, and we hope that this support will be translated to solutions for individuals with this disease, and we build a better society.
I'm Rob Moody. I'm Professor of Public Health at the Melbourne School of Population and Global Health. I'd worked a lot in Sudan, in Cameroon, in Uganda, Mozambique over a number of years, and I was always keen to go back and work in that part of the world. So I went to Malawi. I met the leaders of their School of Public Health and said, look, I'd like to come and volunteer for a couple of years. What do you think about it? One of the challenges in Malawi is obviously that there's a completely overstretched healthcare system. Young doctors do a year maybe, and then suddenly they're the district health officer, the district medical officer. When I was in Malawi, I was really helping to run the Master of Public Health program. One of the things we were able to do as a team was to really increase the effectiveness of the program quite rapidly. I mean, up until then, something like 10% got through in 2 years. We got that up to almost 90%, getting through in 2 years in a very short time, so that it really responded to student needs, and it responded to the university's needs as well. Over the last 5-6 years, we've trained over 500 doctors, nurses, researchers, health professionals. We're also able to start a whole new stream of work on building leadership and management skills. As we know, this is something that's taught in the business world much, much more deeply than it is in the health world. And now, fortunately, I mean, here and I think across the globe, we're starting to realise how important these skills are. This is where, philanthropy can play an enormous role. I mean, it is really that notion of love of the human. And in a sense, it's an expression, I think, of real care that people do give. And it makes things happen that wouldn't otherwise happen. Whether that's in clinical work or whether it's in sort of vaccines and therapies, or whether it's in my area of what we call upstream public health approaches that really do impact on not thousands, but hundreds of thousands of people at a time.